I'm Mette Nicolaisen, and I'm an associate professor here at University of Copenhagen. My main field is plant-microbe interactions and how we can exploit that for better plant growth in plant production.
So we're here at the poster hall at the University of Copenhagen, and I'm sitting here with Professor Nicolaisen. We just finished from her own session, and we're here to ask her a few questions about her session and to know a little bit about her and how she's finding the Congress so far. Let's start with the session. Can you tell us a little bit about the title of your session, and then I give a brief abstract, let's say, of the session, so everyone knows what you talked about?
The title of my talk was the Microbial Ecology in the Green Transition. And I think my main focus was to shed light on the difficulties we do have in actually having what we call the translational power. So translating all the positive findings we find on plant microbe interactions and actually have them working in a real soil environment. So I gave some flash talks on some of the projects that we have been working on and how we have tried to turn our questions around a little bit to try to answer it in a more natural system. And then, of course, also trying to put it out into a real soil environment. It's difficult and it's not an easy task, but I think we have to go that way if we want to make a difference in having microbes helping plants out in the real production of food.
Because as I mentioned before, most of the times we deal with hearing microbes being harmful towards humans, plants, the environment. So it's quite amazing to see how your research is focusing on the beneficial roles. You mentioned in your talk about some limitations of from starting from a pure culture of bacterium and then having it transition into actually the being beneficial. So what are the limitations and what are the stages involved in that?
I think what we talked about was that normally we come from a route where we have an isolate that we can show in the lab on an agar plate that it has some beneficial traits. It can be antifungal, it can be that it provides nutrients, or it can do other beneficial, it can induce stress resilience in the plants. But when we then come out and test it in a real soil system, then it's not working. We simply don't have the power from the systems that we are studying. Mm-hmm. They don't mimic the nature well enough. So even though we can get some information, we cannot transfer it. So the biggest limitation is the complexity in the soil system, because there is thousands of microbes interacting, and they have a chemical language or signaling that they're going, and how that is impacting who is actually ending up on the root surface, helping the plants or being harmful to the plants. That's what we are trying to see if we can figure out. And it's not an easy task.
And you work on soil mainly, right?
Yeah, we work as much as possible in soil, as I was stated. We also do work in pure culture systems because the complexity in the soil is so that specific hypotheses we sometimes have to go from a very simple system and then make our way out in the more complex system. But what we can do is we can learn from the complex systems, going back, trying to disentangle the mechanisms, and then test it back in the soil system to see was that actually the mechanisms that were going on and ending up in whatever feature or phenotype we were looking at.
Yeah. So if we imagine, you have a soil sample and then you do several tests on it, do you find some strains or genuses enriched in the soil that you test?
Yeah, I mean, we had the example also from the lecture where when we actually put in a strain that produces a secondary metabolite and compare that to the same soil system with the same strain, but where it does not produce that secondary metabolite, then we do see that it changes who is actually able to colonize the root surface. And of course, that's not a direct proof at this point, but it at least tells us that having the capability at least to turn on some of these secondary metabolites might have an impact on who else is getting into the root
Yeah.
and colonizing.
Yeah, and now getting into the more fun part of the research. So where do you collect or find your soil sample? Are they Danish-based, or do you collect samples from all over the world?
Currently, we are mostly working in Danish soils, but I have been involved in projects across the world. We have a big collaboration with North Carolina State University. So, of course, we're also getting soil from there, working together with the people over there. Yeah.
Cool, that's nice. So let's turn into the Congress now. How are you finding it so far? I know it's the start, but how do you find being back at an in-person conference after all this time? Is this your first one?
It's actually my second. We had a small one last week here, but this is the first one, bigger Congress that I'm at, and I think it's fantastic. I think it's so good to be back. I think we learned a lot, that we can actually have good seminars and good symposia online, but I think getting in contact with people and having the coffee breaks and talking to them, getting ideas, exchanging ideas, that is needed also. So I'm very happy to be back in person, definitely.
Yeah, and I know that you mentioned a student of yours, a PhD student of yours, is actually giving a poster, hanging a poster here. So what is an advice, let's say, that you could give to someone coming here after quite a while in an in-person conference and talking about their poster?
I think the most important part is to reach out, discuss, get input. Because, of course, it's about presenting what we can do, but it's also discussing how to move on from there, to learn from other inputs, from people who have other knowledge than you have. And only that way, I think we can actually develop our ideas in a positive way. So interacting, talking to people, and also taking have critical discussions about our data. That's the only way we can move forward.
Yeah. Very, very good advice. So yeah, thank you so much for your time and your answers to my questions, and enjoy the rest of the Congress.
Thank you very much.